Hello friends, welcome to Expert Guidance. Today in this video we'll be covering the Unit 3, which is Reproduction and Inheritance of Edexcel IGCSE Biology, and in this video we'll be focusing just on the topic of reproduction. Now in reproduction we'll see what is the difference between sexual and asexual reproduction, how fertilization takes place to form a zygote. In plants, we look over what are the insect pollinated and wind pollinated flower, how plants get adapted to pollination, how fertilization takes place in plants, and we'll also look over what are the different ways in which we can reproduce plant asexually. Now in this video we'll be first covering the structure of DNA. We'll be looking over how the DNA molecule is arranged in the form of double helix, what are the 4 bases present in it, how DNA gets transcribed into messenger RNA, and how it forms a protein. After doing the structure of DNA, we'll come to the genetic terms like alleles, homozygous, heterozygous, phenotype and genotype. We'll see the concept of codominance, the polygenetic inheritance. We'll do the Punnett square with the monohybrid truss. We'll see how sex is determined in humans, and we'll also see how mitosis can help in growth, repair, cloning, and asexual reproduction. In this unit, we'll also cover a very important cell cycle, which is meiosis, which is responsible for formation of gametes. We'll also look over the process of random fertilization and how meiosis leads to variation and eventually leads to evolution. We'll also see the concept of mutation, the genetic mutation, and in this unit, we'll also cover the theory of evolution by Darwin, and we'll also see how we can explain the antibiotic resistance in terms of Darwin's theory of natural selection. Now this is a very important topic, and there are a lot of long marker questions often set out in the exams from this unit. So I would recommend you to watch this video till the end, as we'll be covering all these topics and concepts one by one, and in the end we'll also look over some key terms as well of this topic. So let's begin.
Now reproduction is making more offspring. In a sexual reproduction, it involves 2 parents and involves the fusion of gametes. The offsprings are not genetically identical, and it produces variation and leads to evolution, example human reproduction. On the other hand, asexual reproduction, like that of budding and yeast, it requires a single parent and there's no fusion of gametes. The offsprings are cloned and genetically identical. There's no variation and evolution, example budding and yeast, cutting, runners in plants. Now, when we talk about this reproduction, it involves meiosis. Now, what is meiosis? You can see here, we have one parent cell, and that is dividing to form 4 daughter cells. Apart from making 4 daughter cells, you can see here there are 2 divisions, meiosis 1 and meiosis 2. Second, the chromosome number, we started with 4 and we ended up in 2. So it means that the chromosome number is getting halved. And if you see this picture closely, there's a mixing of the characters called the crossing over. So these are what are the different features of meiosis. It takes place in the sex cells. It is involved in the production of gametes. One parent cell divides to form 4 daughter cells, and the daughter cells are not genetically identical to the parents, and the daughter cells have half the number of chromosomes than that of the parent. It produces variation as it results in crossing over and leads to evolution. Okay, so I hope this concept of meiosis is clear to you. Now, how we can relate it to an actual formation of offspring. When we talk about the male parent and a female parent, they both have 23 pairs of 46 chromosomes. The meiosis happens and it forms a male gametes and a female gamete. With the male gametes, it forms sperms, and with the female gametes, it forms ovum. Both sperms and ovum fuses by fertilization and produce a zygote. Now, the reproductive cycle in a fungi, it involves both sexual and an asexual reproduction. Mostly it is asexual, where it releases the spore germinates and forms the fungal hyphae. On the other hand, in a sexual reproduction, 2 nuclei fuses to form one nucleus, and then it undergoes meiosis to form haploid spore, and then they germinate to form a hyphae. So you should know what is a sexual and an asexual reproduction in fungi. In plants, it involves the asexual reproduction. So asexual reproduction is in the form of runners, stolon, tubers, which produce a genetically identical plant. In sexual reproduction, you can see there's a pollination that takes place, and then there is fusion of the gametes to form the zygote, and that develops into seed. Seed disperse and forms a new plant. In malaria, the life cycle is a little different as it involves a human cell as well as a mosquito. In the liver cell, the asexual reproduction to form the malarial parasite spores happens. In the blood cells, there are the formation of males and the female gametes, and the sexual reproduction takes place in the mosquito, and inside the mosquito, the males and the female gametes fuses to form a spore, and this spores is now ready to be released into a healthy individual whenever a mosquito bites a healthy person, and the cycle continues. So you should know in a malarial cycle what's the role of the liver cells, blood cells, and the mosquito. Next is, it's very important, the DNA. First of all, you should know these terms are very used interchangeably. What's the difference between them? What's a cell? What's a nucleus? Chromosome, DNA, and gene. So inside the cell, we see a nucleus. And inside the nucleus, when we enlarge the nucleus, we see these structures, which are chromosomes. And when we take this section of the chromosome, we see this double helical structure, which is DNA. Out of this DNA, 98% is junk and has no role to play. But 2% of that DNA makes protein. And that segment of DNA that codes for a protein is a gene. Okay, so I hope the difference between these terms is clear to you, and you should be able to define them in the exam. Now, when we talk about the DNA structure, it is composed of a phosphate, sugar, and the phosphate present, the sugar present is deoxyribose. That is why DNA is known as deoxyribonucleic acid. And this phosphate and sugar, you can see it forms a backbone of the double helix. And within them, there are nitrogenous bases, and there are 4 nitrogenous bases: adenine, thymine, guanine, and cytosine. And you remember them as ATGC because A always binds with T with a double bond, and G always binds with C with this triple bond, and this is a complementary base pairing principle. So if we treat the structure of DNA, it's a double helical structure. It's a polynucleotide. What is a polynucleotide? A nucleotide is one component, one monomer, which is a phosphate, deoxyribose, and a nitrogenous base. Many nucleotides, they combine by a condensation reaction and form a polynucleotide, and DNA is a polynucleotide. And one base is making the hydrogen bonds with another base on another strand, and adenine is always next to thymine with a double bond, and guanine is always next to cytosine in a triple bond. And you make sure you remember this: what is a nucleotide, what is the structure, and complementary base pairing principle. Okay, now what is a human genome? Now you can see that there are a lot of base pairs in a human DNA, and Human Genome Project, genome is the complete genetic material of the organisms, and there's a Human Genome Project, which is an international collaboration to sequence around 3 billion bases and more than 20,000 genes. It is important to understand the genetic diseases and the inherited disorders. It is also required to produce better personalized medicine. The Human Genome is important. Genome Project is important to understand evolution and identify the new drug targets. So you should know the importance of Human Genome Project and make sure you elaborate these points. Next is protein synthesis. That's for the higher tier. Now, when you have a DNA, the DNA is converted into the mRNA, and this process is known as transcription, and that takes place in the nucleus of the cell. Then this messenger RNA leaves and reaches the ribosomes in the cytoplasm, and these messenger RNA code is read and an amino acid chain is synthesized. So the DNA copies the code to form messenger RNA by the process of transcription. The messenger RNA leaves the nucleus and reaches the ribosome. The ribosome reads the bases in the sequence of 3 bases, and the transfer RNA brings the corresponding amino acid and forms a polypeptide chain. The polypeptide chain then forms a protein. And always remember, the bases are always read in triplets, and this is known as codon. AUG is corresponding to a codon code of methionine. So the tRNA brings methionine. Then the next is red, which is for phenylalanine. So phenylalanine comes in, and you can see that the polypeptide chain is growing. In this way, a long chain of amino acid is made by reading this triplet, and these amino acids make a polypeptide, and that gets folded to form proteins and enzymes. Okay, so I hope this concept of protein synthesis is clear to you. Now, you must have seen now that DNA forms an RNA, and RNA makes a protein that gives the expression. So what happens if there's a change in the sequence of a DNA? If there's a change in a sequence of DNA, it results in the change in the sequence of mRNA. If the mRNA is changed, the amino acid gets changed. And when the amino acid gets changed, it results in the formation of non-functional protein or enzyme, and that protein will have a different structure, and the active site will be different, so the substrate can no longer fit to it and can be non-functional. This is what is mutation. Now, this can be silent because it results to the change of the base that does not make any change to the amino acid. Harmful if it causes any disease, or beneficial, it gives some extra characteristics to an organism. Okay, so you can see in this, our original amino acid chain is all his, but with the change of one amino acid, the his amino acid changed with pro, and that's what caused a malfunctioning protein. So you should know what is the definition of mutation and what are the changes that a mutation does. That is, it can cause a change in amino acid and a non-functional protein. Okay, and you should be able to explain that in the exam. Next is the genetic terms, because now we are moving on to the genetic process. Now, in genetics, you should know what is a chromosome. So we just have discussed all these four. Chromosomes are the structure present in the nucleus of the cell which holds the DNA. DNA is a deoxyribonucleic acid which is present in the chromosome and holds the genetic information. Gene is a section of DNA that codes for amino acid. Allele is an alternative form of gene. Dominant allele is an allele which is always expressed when present. Recessive allele is an allele which is expressed when present in pairs. Homozygous alleles, when both the alleles are same. Heterozygous are when both are different. Genotype is a genetic makeup. Phenotype is the physical characteristics expressed by a genotype. A Punnett square is a square that shows the possible genotype from the parent gametes. Now let's take an example of a genetic cross. Now, let me show you a genetic cross and how this genetic cross can be used to predict what would be the various components, what will be the various combinations of the offspring. And in the exam, you should be able to write what would be the genotypic ratio and what would be the phenotypic ratio. Okay, so let's have a look into from these genetic terms using them. So, for example, we are crossing a heterozygous yellow plant with the green plant. Now, yellow is dominant, which is capital Y, and green is recessive, which is small y. So, a parent would be capital Y, small y, which will be yellow in color, and small y, which will be green in color. Now, when we are crossing it, these two will separate. So, the gametes will be small y, and big Y and small y. Now, in a Punnett square, we'll do a cross. So, it will be capital Y, capital Y, small y, small y, small y, small y, small y. So, what ratio did we get? So, here we got 50% to be green and 50% to be yellow. Okay. Now, let's take another example. Here, our brown eyes is dominant, which is capital B, and small b is the blue eye, which is recessive. Now, we are crossing homozygous brown eye with the heterozygous brown eye. In this case, we got all brown. So, the phenotypic ratio is 100% brown. But if we see the genotype, it is 50% homozygous and 50% heterozygous. Okay. So, this is how we do the cross. We take the parent, separate them into gametes, and do the cross. For example, in the second question, we are doing homozygous A, homozygous dominant, and homozygous recessive. We got all 100% dominant. On the other hand, if we took heterozygous with the heterozygous, in this case, we get 75% to be dominant and 25% to be the recessive allele. Okay, so I hope you are clear with how you need to do these crosses, and these can come in the exam where they can ask you to predict the offspring phenotype or genotype. Now, in the similar way, we can do a sex determination. Now, in humans, you need to remember that the male is always XX and female is always XO. So now, and we draw the Punnett square of that, the male gametes, it will separate, and it will separate into XO, the male gametes, and the female would be XX. Okay, so what are the possible combinations we can have? We can have XX and X and O, and then again XX and X and O. It means there are 50% chances of getting a male and 50% getting a female, and to determine the sex, it's the male that is responsible, because female always gives the X chromosome. Now, it depends on the male. If the male gives X or it gives X naught or XY, that will determine whether it's a male or a female. Okay, now let us look at what is polydactyly. There are two disorders which are there in your syllabus. One is a polydactyly, another is a cystic fibrosis. Now, polydactyly is caused by a dominant allele. Now, what is the meaning of a dominant allele? Dominant allele means that even if one of the parent has an allele, the offspring has a chance of having it. So you can see in this, one of the parent was having the allele, so there was 50% that was affected. On the other hand, cystic fibrosis is a recessive disorder. Now, for a recessive disorder, both the parents should have an allele to infect the offspring. Here, both father and mother are carrier. They had one allele, and that made 125%, that is a quarter of the offspring, to have cystic fibrosis. If only one would have that disease, the offspring will not have. They could be the carrier, but they'll not be a sufferer. Now, cystic fibrosis is a disease which there's a sticky thick mucus that is developed, and it blocks the airway, reproductive tract, and digestive tract. Now, what you need to remember here is, in the dominant inheritance, one of the parents should be affected in order to make the child affected. But in a recessive, both the parents should have the allele in order to make the child affected. Now, in the exam, they can give you different combinations, and they can ask you to make the cross and predict what are the percentage of the offspring that will be affected. Now, since we know there are a lot of genetic disorders that are there, there's another thing which is very hot these days is genetic screening. Now, what is genetic screening? Genetic screening is we basically screen the genes for the unborn child to find whether the likelihood of getting a disease. Now, in genetic screening, when we screen the genes of the unborn child or the fetus, we can find whether the child is suffering from any diseases or not. Now, how we do that? There are three methods. One is amniocentesis. You can see in an amniocentesis, we take out the amniotic fluid that is surrounding the fetus, and it has some fetal cell, and then we screen that for genetic disorder. Now, this has to be done in a later stages, around 16 to 20 weeks, whereas chorionic villi sampling can be done in an early stages, and it involves taking the fluid from the placenta, which contains the fetal cell. And then we have the IVF screening. In IVF screening, these are for those couples who have taken in vitro fertilization as a method of fertilization. In that, we can screen the embryos before inserting them into the uterus. Now, these techniques are very important because they can provide the couples with the genetic counseling and helps to diagnose any problem, take any measures, or to terminate the pregnancy before the child is born. Now, a very important concept, variation. Variation is different amongst the individuals. You can see in your class, you can see there are some students who are long height, some students are short heighted, some students are already fat, some are thin, some are skinny. So the variations can be of two types, continuous and discontinuous. Continuous is mostly it is environmental triggered, like height and weight. So if I try to match the height of the individuals of your class, I can get any value. I'm not certain what the height would be of all the individuals, because since height is an environmental factor plus genetic, but since environmental influences, it takes any value. On the other hand, if I try to plot the different blood groups which are there in your class, I can only get 4 sets, A, B, A, B, O. Because since blood group is genetics, it can take fixed value. And this kind of variation is known as discontinuous variation. So discontinuous is genetic, whereas continuous is environmental. Continuous can take any value, where discontinuous can take discrete values. Now, since we know there is a variation, there's something called the natural selection. Now, according to Wikipedia, natural selection is the differential survival and reproduction of individuals to difference in phenotype. It is a key mechanism of evolution, the change in the heritable trait characteristics of a population over genetics. Now, what does that mean? There was a scientist called Darwin. He expedited a lot and he found that every organism has an inherent capacity to reproduce. Despite of that, the population remains constant because of the very important phenomenon prevailing called natural selection. The nature puts the selection pressure, and those who are suited for the selection pressure, they get a chance to survive, reproduce, pass on the traits to the next generation, and results in evolution. Okay, so you can see this was an ancestor finch and it gave birth to 3 different finches and they were exposed to the different environments. You can see now they all have different beaks. Why is that? Because this finch was growing in that where only insects were there as source of food. The second finch was growing where there were only woodpecker type or insect eating. And the third was the seed eating. So the ones who has to take the seed as little brown out, on the other hand, the other two had very sharp. And that is different from the ancestral finch. Why is that? Because all these 3 exposed to a different environment, they had different natural selection pressure. The one that has the characters to fit to that, it survive, reproduce, pass it trait to the next generation, and now we have the different evolved versions of the finches. We can take another example of the drug resistance in bacteria. If you give drugs to any bacteria, the bacteria will die. Now, only some bacteria is mutated, like these 2, and they were resistant to that particular drug or antibiotic. So what happened eventually? All the other ones died and only these 2 survived. And when these 2 survived, they got a chance to reproduce. And when they reproduced, they multiplied and passed on their traits to the next generation. So how it works? There's a mutation. Mutation leads to variation. Then the nature selects which is the best variation of which is the organism which has the best adaptation. So there's a survival of the fittest and that leads to evolution. Okay. Now, let's take a very important term called selective breeding. Now, selective breeding is what has been done since ages, where we artificially mate the 2 desirable characteristics parents to reproduce to form the required offsprings, and then we select the one with the best characters. For example, we can produce high milk yielding cow. We can produce disease resistant high yield crops. Now, this is a slow process, time consuming, causes reduction in a gene pool. Undesirable traits can also get selected, and a disease, if happen, can result in the elimination of the entire population. So selective breeding is harmful in this case, that it reduces the gene pool. So now we have a very new emerging technology, which is genetic engineering. Now, genetic engineering is altering the genes by inserting the desired genes to produce genetically modified organism. Now, how this works is we select a foreign DNA, which is a gene of interest, and we isolate that gene of DNA, and then we take a bacterial plasmid, and then we cut the bacterial plasmid so that we can join our plasmid with this our gene of interest to make a recombinant DNA. So first we isolate the gene, then we cut the vector and the gene, and then we join the 2 to make the recombinant DNA. Then we insert this recombinant DNA into the bacteria, and that is known as transformation. And then we select that which is the bacteria that has now got transformed with our foreign DNA. We select it and make multiple copies of that particular bacteria. In this way, the bacteria will express our gene that we require. So what are the applications we can have with this genetic engineering? We can make that tomato with a greater shelf life. We can insert the gene for vitamin A in the rice and make golden rice, which is rich in vitamin